Hello and welcome to today's webinar. My name is Astrid Gall and I'm an outreach officer for Ensembl. We will be looking at genes and transcripts in Ensembl in this webinar. This is the outline of today's webinar. I will explain different gene views in Ensembl. I will explain the different annotation methods that we use. I will talk about some indicators for quality of transcripts, such as golden transcripts and the GenCode and main transcript sets. And this will bring me to the question of which transcript you should use if you only wanted to use one. And towards the end, I will also introduce our system of stable IDs and gene ontology. Okay, so this slide shows a typical gene view in Ensembl. We are looking at the BRCA2 gene, which consists of multiple transcripts. Transcripts is the term that we use in Ensembl. You may also hear splice variant or maybe isoform if you're used to working with proteins. They essentially mean the same thing. So we display our transcripts as a combination of these blocks, which are the exons, and these lines, which are the introns. And we color code them. We have red for protein coding transcripts, blue for non-coding transcripts, and then we have transcripts which are shown in a golden color, and these are the so-called merged transcripts. And they represent high quality transcripts, and I will come back to those a bit later. The exons are colored in if they are coding, and they are not colored in if they're non-coding, and the same applies for the UTRs or untranslated regions. Also, from this kind of view, you can tell on which strand the gene is encoded. You can see that if you have these labels, these names of the transcripts with a little arrow next to them, if they point to the right, that means forward strand. If they point to the left, that means reverse strand. Also, our introns are pointing upwards, such as little peaks. Again, that means forward strand. If they point downwards, that means reverse strand. And finally, for BRCA2, you can see all the transcripts above this blue line with the contigs here. And that again means they're on the forward strand. If they were below, that means they are on the reverse strand. We use two different annotation methods in Ensembl. There's the automatic annotation, which is done by the Ensembl team, and this is carried out for all species that we have in Ensembl. And on the other hand, there's the Havana team, which does a manual annotation. And this is only done for 4 species, namely human mouse, rat, and zebrafish. For the automatic annotation, we use experimental data that comes from a variety of databases to make genome-wide predictions for all the species in Ensembl. So this is completely automated. However, there is real biological data behind this. And this is the biological evidence that we use for our automatic annotation. We have data from the INSCC, which is the International Nucleotide Sequence Database collaboration. This is the collaboration between ENA at EMBL-EVI, the DNA database of Japan, and then NCBI in the United States. And we use the cDNAs, the ESTs, and the RNA-seq data from them. We also use protein evidence for our annotation, and this comes from UniProt. On the one hand, we use manually curated proteins from SwissProt, and on the other hand, also unreviewed translations from Ensembl. And finally, a lot of our gene annotations are done based on homology to other species. So for example, the majority of the genes in the chimpanzee genome is annotated by mapping from cDNAs or proteins of the human genome. Now, the manual annotation is very different. This involves a small team of people who actually look at the underlying raw data and read the literature on specific genes and transcripts, and they then make informed decisions on a case-by-case basis for individual transcripts. They again do this on a genome-wide basis, but there's a lot of work involved there. So obviously the manual annotation is a lot slower than the automatic annotation, and therefore we can only do it on a smaller scale. However, it also has several advantages. First, it tends to be more comprehensive, so we get more transcripts per gene and also more genes per genome typically. And it has in particular value for regions of the genomes which are very difficult to annotate by automatic methods. For example, UTRs, splice sites, single exon transcripts, and also highly diverse regions such as immunoglobulins. For a small number of species in our main site, Ensembl, we also have imported annotation. This imported annotation comes from external sources that we trust and that carry out rigorous quality control. This mainly applies to our model organisms. So we have fruit fly annotation from flybase, C. elegans from wormbase, and then yeast annotation from SGD. More recently, we also imported annotation from a company, Horizon, for the Chinese hamster ovary genome, and then annotation for a couple of mouse species from UCSC. If you work with a non-vertebrate, all of the annotation is imported into Ensembl genomes. So this is our sister site you will have heard about in the webinar on Tuesday. And this Ensembl Genomes sister site covers our non-vertebrate genomes. And as I said, all the annotation there is imported. Okay, so now I'm coming back to my golden or merged transcripts. These are the transcripts where our two annotation methods, namely the Ensembl and the Havana annotation, agree. You can see the example for the BRCA2 golden transcripts on this slide. Naturally, as we only do manual annotation for 4 species, we only have golden transcripts for these. However, these golden transcripts are transcripts that we consider to be of high confidence and quality for the reason that these methods agree on them. So this brings me to the GenCode gene set. The GenCode gene set is available for human and mouse, and it is released on the same release cycle as Ensembl. And it is the merged gene set of the Ensembl automatically annotated genes and the Havana manually annotated genes. And this gene set is used by many large projects, including ENCODE, GNOMAD, 1000 Genomes, and GTX, which you may have heard about or may be working with, in fact. We are, as EMBL-EBI and Ensembl, we are aware that there's also NCBI who do annotation. And in the past, it has often been a problem that the annotation for specific transcripts between the NCBI and EMBL-EBI did not agree. So we have been working with NCBI to solve this problem. And you can now access a new transcript set, which is called main transcripts. And this stands for matched annotation from the NCBI and EMBL-EBI. These are transcripts that have 100% identity between the RefSeq, so the NCBI transcript, and the Ensembl transcript, and they are aligned to GRCH38. We have released a first set of these, which are called main select. Here we select one transcript for every protein coding gene, which is agreed to be the most biologically relevant transcripts for that gene. And these main select transcripts are currently available for about 2/3 of the protein coding genes. And this is work in progress. So talking about various transcripts and how they look in the browser, you may have the question of which transcript you should use. If you only wanted to use one, for example, to clone it, and do some expression analysis, something like that. So this is the recommended approach here, summarized on this slide. You start by looking at different criteria at the top. So first you check whether there's a main select transcript for your gene. If there is one, then you choose this. If there's none, then you go to the next step here, looking at Apres principal isoforms. Apres is a group who annotate major isoforms based on protein structures and cross-species alignments, and they give scores from P1 to P5, where 1 means the best. And furthermore, so if you have two transcripts, for example, which both have an Apres 1, you can go to the next step and you check if there's a GenCode basic transcript annotated. GenCode basic means it is a GenCode transcript which is complete on both the 5' and the 3' end. Furthermore, step number 4, you can also look at the transcript support level, or TSL. This again ranges from 1 to 5, where 1 is the best. And then you also have information about the consensus coding sequence. This is a kind of a precursor of this main project, where NCBI and Ensembl EBI agreed on a matching coding sequence only. And finally, you can look at your golden transcripts, which stand for matched annotation from Ensembl and Havana. So take-home message from this slide is you go through these different criteria from the top to the bottom and use them to choose a transcript. I will give you some examples now. Here you can see transcript tables for 3 different genes. They include information about the name and the transcript IDs, its length, and then in these columns here, highlighted in red, we have the biotype. So this refers to the colors that I explained at the beginning, where this golden color means merged transcripts. Then we have the CCDS here, and on the right-hand side we have flags. And here you find main select, Apres, GenCode, GenCode basic, and TSL. So looking at this gene now, if you wanted to only use one transcript here, you would choose the main select transcripts just at the top at the top of the table. Now for the second gene here, you can see there is no main select transcripts, so you would need to look for the Apres principal isoforms, and there's one transcript here that has 1 annotated, and therefore we would choose, in this case, the second transcript in the table. And finally, in my last example, there is no main select transcript. 2 have Apres P1, 2 have GenCode basic 1, which leaves us to look at the transcript support level. And you can see one has 1 and another one has 5. And therefore, in this case, we would be using this transcript here. Okay, so now a little bit of background about our system of stable IDs that we use. So if you have worked with a gene over a long time, for example, sometimes it happens that your gene name changes, and this is a problem. And to circumvent this problem, there's a system of stable IDs. So these are exactly what they say. They are stable, they don't change over time, and they're also unique. And they take a certain format in Ensembl. So they, in Ensembl, our stable IDs always start with an ENS for Ensembl, and then we have a G for a gene, a T for a transcript, P for protein, and so on. And that is followed by a number. If you don't work with human, you also see an additional 3 letters between the ENS and the G, T, and so on, which represent your species. So here, for example, you have a DAR for zebrafish. If you wanted to find out more about our gene annotation system, please have a look at this publication here. So now I'm gonna spend a couple of minutes to talk about gene ontology before I go into the live demo. Gene ontology is a system that we in short just call Go. Why do we need this? Well, if you talk to somebody about the function of their favorite gene, they may say it's involved in innate immunity. And the next person you talk to says it's involved in non-specific immunity. And the next person says it's involved in cytokine response. So on. There are a couple of problems here. One is that different terms are used for the same thing, in this case a function. And the other one is that the gene descriptions tend to be very specific. Here is where gene ontology comes in to help us to ensure that we all talk about the same thing. These Go terms form a controlled and searchable vocabulary. So here I have an example of a Go term, innate immune response, which in fact has an accession number, which is shown in bold here. And then you can look up this term and you get a very detailed description of what this term actually means. And this ensures that everybody knows what you're talking about. And we get these terms from geneontology.org. The second thing about Go terms is that they are hierarchical. So in my example here, the innate immune response is in fact a daughter term of immune response, and it itself is a parent term of all of these other terms related to the innate immune response, including things like response to interferon gamma or virus gene silencing. So again, you can go into gene ontology and browse this hierarchy of terms. Okay, so this is the end of my presentation. I'm now going to do a live demo where we will look at the UQCRQ gene and find some information about this gene and its transcripts. And you are welcome to follow while I'm explaining this, of course. Okay, I'm leaving my presentation and I'm going into the website ensembl.org. This should look familiar for you if you have done the webinar on Tuesday. Now I'm searching for a human gene which is called UQCRQ. In my search results, I can see that my top hit is in fact this human gene. So they are ordered by kind of how often these pages are accessed. I have restricted to human already, but I can also go to another species, and I can also restrict to categories such as genes, transcripts, and phenotypes. I will click on the top hit, the human gene now, and this takes me to the gene tab. Make it a bit smaller. Okay. So Ensembl is a tab-based browser. The location tab that you were looking at earlier is always open, and we're now in the gene tab. All of the menu items on the left-hand side are now specific to gene displays. On the gene tab at the top, we have our gene symbol, and we have got information where it comes from here. It's from HGNC, the Human Genome Nomenclature. And we have our Ensembl stable ID next to the gene symbol. You can also see information about the location of this gene on chromosome 5 on the forward strand, and the fact that it has 6 transcripts, which you can see in the transcript table here. You can hide this table and show it again. And towards the bottom of the page, you can then see the graphical information about the transcripts of this gene here. I'm going to explore a few things about this gene via the menu on the left now. And the first thing I will go to is the sequence view. So I click on sequence in the menu on the left. And what I get here is a FASTA sequence of this gene and some surrounding region. So if you're not familiar with the FASTA format, Just to explain this briefly, it has a header which starts with a greater than sign. So in this header here, we include information about the genome assembly, the chromosome, which is 5, the start position, the end position, and the strand, which is 1. And 1 means forward strand. All of this is separated by colons. And then you have the actual sequence in the letters here. We also highlight some features of this sequence in colors to help you with it in the browser here. So we have, as you can see, the UQ-CRQ exons in red and bold. So this is a UQ-CRQ exon here, and all exons in this region are shown in yellow. So this, for example, is a neighboring exon. Why do we see this? Well, because we're showing some flanking sequence here as well. And if I don't want to see this, I can go to configure this page in the menu on the left, and then set the flanking sequence to 0. What I can also do is I can switch on variant displays here. And some of you may see these in the browser right now. These are the colored highlights behind the nucleotides. I will leave it like this because we come back to the variant views in our variation webinar. So I'm just closing this menu now and point out more things. One thing that you can do is to blast parts of this sequence. And for this, you just highlight them with your mouse, and you can drop the sequence into blast directly. And you can, of course, download this sequence as well by using this blue download button here. You have options to download sequence in FASTA format, which I just described, and this is very useful for bioinformatics downstream applications. And you can also download the sequence in an RTF format, which includes the colored highlights that I've just described. And this is a format that you can use in a word processor, such as Microsoft Word, and that a lot of people use, for example, for primer design. You have options to select what type of sequence you want to get here as well. I'm going to close this now again and go to the next menu item where we look at the ontologies. On the left-hand side here, under ontologies, you have 3 Go sections. There's one for Go biological process. And this summarizes what the protein does. Then we have Go cellular component, which summarizes where it is located. And we have Go molecular function. This is how it does whatever it does. So for all of these, you have a table such as this one here for the functions that lists the Go terms and accession numbers together with their associated transcripts. So please note that our Go term assignments are always based on the transcript IDs, which are listed in this column of the table here. You can then explore the terms in more details in gene ontology. So if you click on this link here, for example, and open it in a new tab, it takes you to Quick Go, an Ensembl EBI resource, where you can find more information about this Go term and where it sits in the hierarchy of other terms. Okay, I'm going to close this and go back into Ensembl. And I will click on gene expression towards the bottom of the menu now. On this page, we display data and, in fact, the interface that we get from Gene Expression Atlas, which is also an Ensembl EBI resource. You can see the link here at the bottom. So you have a human body map on the left-hand side, which you can change from a male into a female body. And you can also change this into a brain, if you're interested in brain tissues. And in this body map, you can highlight different tissues and organs And these then light up in the heat map on the right-hand side, and the other way around as well, if you mouse over these. In the heat map, you can see the different tissue types at the top, and then here on the left-hand side, from top to bottom, you have different experiments where this data comes from. And this is all imported from Expression Atlas. What you can do in this heat map is you can mouse over these lines and you get the actual expression levels in TPM. You can also sort the heat map by expression rank, or you may filter for your organ of interest or an anatomical system of interest by deselecting things and selecting things here. And there's an option to download this, of course, as well. Gene expression is an example of external data that we import. And you can go to external references further down in the menu on the left to see other databases where this gene is mentioned and where you can find out more details about this gene. One is Expression Atlas, but we also have HNC for the symbols, MIM, which is in Mendelian inheritance of men. We will talk about this in the variation part, and some others. And here you can then go to these other databases and find more about your gene. Okay, so now I'm going to scroll to the top of the page again and show my transcript table. So if we look at this transcript table here, and we would need to choose just one of these 6 transcripts, we would choose this main select transcript here. So the first one in the table, which is also the golden transcript. Therefore, I now click on the transcript ID in the table, which opens a new tab, and I can hide my table again. Yeah, so you've got your graphical overview of this transcript as well. And you can see that it has 3 exons here, which is also set at the top of the page. Now let's explore the biological evidence that was used to annotate this transcript. You can do this for all the transcripts in the table here. And for this one, I will now click on supporting evidence in the menu on the left. So in this view here, you can see our transcript at the top in a golden color. And then the tracks underneath here, they all represent types of evidence that were used to annotate this transcript. And this can vary between the exons and between different transcripts. We color code this evidence. We show cDNA evidence in green, protein evidence in yellow, and EST evidence in purple. And you can click on the names of the tracks and also on individual blocks here to find out more about the evidence that was used here. So this, for example, is a UniProt record, so protein. And I can click on that ID here to go to UniProt, where I can read more about this protein and also have a look at the annotation score here. So UniProt also annotated this protein with a score of 5 out of 5, which is really good. So this is a high quality annotation. Okay, I'll go back into Ensembl. I will now show you a sequence view on the transcript tab. On the transcript tab, we have 3 sequence views. One for exons, one for cDNA, and one for proteins. And I will demo the one on exons and leave it for you to explore the cDNA and the protein view. So I'm going to click on exons now. If you remember from earlier, this transcript has 3 exons. And these 3 exons here are now listed in this table at the bottom of the page. They're numbered 1, 2, 3, and they have a exon ID. You can find further information about these exons and also upstream, downstream, and intronic sequence in this table in these columns here, and of course a sequence on the right-hand side. The sequence again is color-coded. We have flanking sequence, so starting with the 5' upstream sequence in green. We have UTRs in orange. We have translated sequence starting with this ATG here in blue. And then we also have intron sequence in gray. Again, similar as on the gene tab, you can customize what you see here by clicking on Configure this page and decide how much flanking sequence you want to see, if you want to display full introns, if you want to see variants, and so on. I will leave everything as it is and just say that the BLAST works in the same way here as for the gene tab, and you can download the sequence here using this button, again in the same formats, and you can decide what type of sequence you want here. Okay, we also have links to external references for transcripts, and these come into different parts. We have the general identifiers, where you can find links to these transcripts in other databases, and this includes CCDS, ENA, and also RefSeq. If you're used to working with RefSeqs, this is where you can find the RefSeq IDs. And there's also external references to oligoprobes. And this is something I won't show, but it's something included in the exercises, so you may want to have a play with this later. Okay, so finally, I'm going to show you some protein views, which are in this section here, protein information. I will go to protein summary first. In the protein summary view, I can investigate how my Ensembl proteins with their exons map to different protein domains from other data protein domains, which were annotated by different methods and imported from different databases, such as PFAM and Panther. And you can see here we have this cytochrome B domain here, and most of these methods agree, but not completely in terms of the coordinates. If you are somebody like me who prefers tables over graphical views, you can go to Domains and Features, where you get the same information in a table format, plus more details about the start and end coordinates and links out to these other databases with the protein information. Finally, we also have 3D protein models for many, but not all of our transcripts. We import these from PDBE. This is the Protein Data Bank of Europe. We use this viewer that they have to display our proteins. I'll make it a bit smaller to bring this legend here into view. So this is an interactive viewer. You can kind of turn around the proteins. You can highlight different parts of them. So what I'm going to do here is I switch on exon views by clicking on this eye icon here. And now my two exons are highlighted. I can also do the same for protein domains, such as PFAM domains, and later for variants too. So here you can find out the effects of the variants on the proteins here. And yeah, this is all actually what I wanted to say about this view. We have these for the protein structures which are available in PDBE, and this is not the case for every single gene in Transcript. Okay, so this was the end of my live demo. I'm now going back into my presentation and spend a few minutes talking about what we will do next and point you to some more resources. So our next webinar is on Tuesday, the 28th of April. where my colleague Michał will talk about variation data and the Ensembl web. So we import variation and also phenotypes from a number of different resources. In his webinar, Michał will talk about how you can find variants in genes and regions and access additional information, including population frequencies. This should in particular be interested for people who work in clinical settings or who work in breeding programs, things like this. We will also introduce our first tool, the variant effect predictor, which is very powerful and allows you to analyze your own variation data for the effects on the genes. This webinar will be on Tuesday, 4 p.m. British summertime. So now I would like to encourage you to continue asking questions in the chat box. I hope you have done this already, and Ben and Michał answered so far. We will keep it open for a while and just be there and check if there are more questions coming up. I would very much encourage you to have a look at the exercises after this webinar, which you can find in the online course. There are solutions as well. If you want to have help with the exercises, please refer to the solutions in the online course or ask questions in our living document where we are available to answer them. And you can also email us at helpdesk@ensembl.org to ask direct questions to us and get a response directly. So finally, here are a few more resources about Ensembl where you can continue your own learning. We are part of EBI training, so this is where you can find a lot of courses, including this one online. We also have our own page with tutorials. We have a YouTube and a Youku channel where we will upload this webinar video and where you can find a lot of other videos as well. You can email us. You can join one of our public mailing lists if you're interested in new data and new releases. You can join our announce email list, which is low traffic. And if you are a programmer and want to find out and get help with programmatic access, you can join our dev mailing list. You can follow us on social media. We have got a Facebook account, Twitter accounts, and also a blog, which is at ensembl.info. We do write publications, which you can find under this link here. It's a long list. We write a yearly update on Ensembl and on Ensembl genomes in nucleic acid research, which are the main yearly papers, but there are also topic-specific publications, which you will find on the website. Please do cite us when you use Ensembl. We are a freely available resource, and it's important for our continued funding and for providing this service to you to get citations as well. And this is us. Last year, we are a group of about 70 people. This leaves me to thank the entire Ensembl team, which you can see in names here. I'd also like to thank our funders. Our main funding comes from the Wellcome Trust, and there's funding from other funding agencies, which you can see at the bottom of this slide. So yeah, thank you very much for attending today's webinar.